Hi, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today I am excited to welcome our guest, Dr. Jakob Mørkeberg. Listeners, Jakob is Senior Science Manager at Anti-Doping Denmark. And for more background on Anti-Doping Denmark, make sure you check out our recent episode 122 of the show. But today, we are going to go more in-depth talking about Jakob's research and development efforts to advance dried blood spot testing, as well as other areas of the field of anti-doping. So, Jakob, welcome to our show today. How are you?
Thank you. I'm doing really good. So thanks for having me.
Well, we are excited to learn more about you and the wonderful work that you do in our interview today, but perhaps you could start by telling us more about your roles and responsibilities there at Anti-Doping Denmark.
Yeah, sure. So actually, I started working for Anti-Doping Denmark for now it's already 11 years ago as a scientific consultant. And actually, the first half year was a half-time position because I did some research at the same time at a hospital. But after 6 months, I was offered a full-time position here at Anti-Doping Denmark. And the main objective at that time for my position was to implement the blood passport in Anti-Doping Denmark. So I was actually doing research into the blood passport at the hospital. And then they offered me a position where I could kind of implement that in Anti-Doping Denmark. So the biological passport has been my main working task since then. But after some years, we created a new setup around our research committee. So I'm now the chair of that committee, and I've been that for 5 or 6 years now. And actually, I'm also been involved in the testing strategy and also in an I&I work. So it's become quite a broad role with many different tasks, which is very interesting for me in my daily work, I think, because I also work with a lot of different colleagues here in Anti-Doping Denmark within the different areas.
Oh, very cool. And you mentioned, of course, that you are chairman of Anti-Doping Denmark's research committee. Can you tell us a little bit more about this committee and what it does?
Yeah, sure. Actually, it was created before I arrived. And at that time, it was consisting mainly of external professors within different kind of areas, to be medical, social science, stuff like that. But then it was kind of restructured a bit. So we wanted to get more committee members in from within the secretariat and also from the board, who had, I think, greater knowledge of what the actual anti-doping problems were that we were facing in our daily lives. So that was created. So we're now 8 members of the committee, both with people from the secretariat, people from the board, but also we have a couple of external members as well. And we meet 3-4 times per year. And it's mainly discuss the research areas that we wanted to focus on and that external institutions can apply money for such research areas. So that's our main objective. We also now and then host research symposium or conferences, which we also kind of make come to life from the research committee.
Excellent. And you mentioned you've been there at Anti-Doping Denmark now for 11 years. I think time goes by quickly and that it started with this consulting position, but perhaps we could back up a little bit further. Can you tell us, Jakob, what first got you on this path towards anti-doping?
Yeah, so it's been a long journey. I've always been really interested in sports and such, both watching in the telly, but also doing a lot of sport myself, a lot of football, running, stuff like that. So quite early, I kind of decided to educate myself within this area. So first I took a bachelor's degree in sports science, and then on top of that, a master's in human physiology. And here I was interested in really which factors that limit sports performance in humans. And then when I had to choose a topic for my master thesis, there was this ad coming up where a research group at the Copenhagen Muscle Research Center were looking for master students to help out with actually a blood doping study. So old-fashioned blood transfusions where you blood dope with your own blood. So actually they wanted to examine how the blood flow was regulated in the working muscle and how the manipulation of oxygen impacted that blood flow regulation. So one aspect of the study, which I found very interesting, was also to test the subjects on their performance on a cyclogometer to examine the performance-enhancing effect of blood doping, but also especially looking at different biomarkers as a kind of an indirect test strategy to detect such blood transfusions. And actually, these biomarkers, they're the one we're using today in the blood passport. So after this study, I continued with a PhD in medicine, also focusing on if we could detect autologous blood transfusions, meaning blood transfusions when you do it with your own blood, and whether we could come up with a test for this. So that was how I got into this whole anti-doping area.
Very interesting. And I know you've been there for some time, and as you sort of alluded to, priorities of the organization from a scientific perspective have shifted a little bit over the years. Can you comment on just how Anti-Doping Denmark's scientific focus has maybe evolved or grown over the years?
I think in the beginning, as I just mentioned, the research committee consisted of these external members, mainly professors, and they were excellent in their daily research activities, but maybe they were a bit far from the anti-doping work, their daily life, and what problems the anti-doping community were facing. So as I said, we did this overhaul of this whole structure within the research committee, bringing in more staff and board members who had different areas of expertise and knew which problems we were facing on a daily basis. So after that, you can call it revision of the committee. I think we've become quite good actually at looking how practical problems that we are facing in our daily activities can be solved through new research projects. And we have become better in granting money to research projects that we think will have an impact on our daily work. An example of this is the dried blood spots, which I think we will talk more about later on, which we have actually been working on for several years now. We have also focused a bit on the prevalence of doping. So how many people are actually doping, not only athletes, but also people in the community. So we have granted some projects to look at that, put quite a lot of money into research developing the athlete biological passport. But we're also focusing on the health effects on, for example, the anabolic steroids. So more kind of within the society, what kind of side effects there when you take these doping substances.
Certainly. I think this public health element of anti-doping is very critical as well. And you mentioned dried blood spot testing. I think this method is already having big impacts in anti-doping. And Anti-Doping Denmark has actually been part of the efforts to develop and test these dried blood spot or DBS methods over the years. So Jakob, can you talk about how Anti-Doping Denmark maybe first got involved in these projects and what have been some of the key milestones for you over the years?
It actually has been quite a long journey, I would say, already. I heard about the method at a USADA meeting. That's these annual science symposium, and it might have been maybe 8 or 10 years ago, where the former CEO of Anti-Doping Switzerland kind of mentioned the method and had looked into it. And actually, I found it very interesting and saw some prospects with this method, maybe especially within the fitness sector. It's a little bit special in Denmark. We both test in sports, but also in the fitness sector. And here, I would say the environment during the doping control is a bit different. We have a lot of positives in the fitness centers. So providing a urine sample is not very convenient for the subjects who are notified for a doping control, and they are pretty reluctant to, sometimes at least, to collaborate with the doping control officers. So there was some prospect with a method that relied on a blood sample instead of a urine sample. So the idea was first, of course, to learn the collection method, but also get the water laboratory in Oslo. We don't have our own water lab in Copenhagen, so we send all our samples to Oslo. So we also had to get them on board. If we have to use the method, they needed to have the analysis capacity in place. So together with the actually researchers at the Copenhagen University and then the Oslo lab, we applied at something called the Innovation Foundation in Denmark for an industrial PhD position. And we already had a good candidate for that PhD, Sara Solheim, which I know has been mentioned a few times in previous episodes. She's already been doing some projects for us previously. So we wanted to focus on the detection of different substances that we knew were frequently used in the fitness sectors, such as testosterone and clenbuterol. So the objective was actually to get some real excretion data from human subjects and, of course, furthermore, to develop the analysis method in the Oslo laboratory. So that was kind of the first part of the journey. The second part was that we wanted to get the screening in the lab, a good screening method, which captured, I would say, the most frequently used doping substances out in the fitness centers. So we wanted to expand the analysis that was developed during Sarah's PhD to get a larger number of analytes into our analysis menu. So again, we went back and looked at the most frequent used doping substances that we had found in the urine samples for the last 2 years in the fitness centers. And when that was implemented, the next step was to try this DBS approach in real life, actually, and compare the analysis with the more conventional urine analysis. So we kind of had to, in some way, see, do we find the same substance in urine than we do in the DBS? So we actually go out in the fitness centers. And since it was a research project, the ones that were notified for the doping control, they had the choice of also providing a DBS sample at the same time. So we had duplicate samples. We had a urine together with the DBS in many of the cases. And this study has just been published, and the conclusion was that we had a 95 correlation between the urine samples with the DBS. So that was quite successful, I think. And therefore, we decided to actually move from the conventional urine collections in the fitness centers to the DBS collections in January this year. But I have to say that usually the detection window for these substances, mainly anabolic agents, they are greater in urine than in DBS. But it's a little bit special in the fitness centers because the athletes tend to take quite high dosages because they really aim for these large effects on the muscle size. And that makes it easier to get a positive in a DBS. And then the next step now is actually just to expand the testing menu. So we funded a new project last year with the objective of getting more analytes into the screening menu in the water lab in Oslo. And this study is underway now.
Oh, that's really exciting. I have to ask, when you came in to gyms for the first time with this research study that you were proposing to people who might not be familiar with research, might not be familiar too much with the anti-doping methods, was it difficult to convince people to provide the second sample of a dried blood spot in addition to the urine testing that they maybe already didn't want to do?
I don't exactly remember the success rate, but obviously there were people who were not wanted to do it. We had a small giveaway, so maybe that helped a little bit. But actually, looking at the numbers, we had 27% of all the urines that were collected were positive, and almost the same in DBS. So actually, there was quite a great amount of the people that were notified that actually wanted to provide the DBS sample as well. So yeah, maybe also a little bit of surprise to me actually, but...
Oh, very interesting. And I think this was an important study and an important step in general to just make this process a little bit smoother. Like you said, there's some specific challenges involved in collecting urine in these anti-doping testing environments. So what have been maybe some of the challenges or hurdles or maybe even benefits that you went through in order to really actually implement this new approach of dried blood spot testing in anti-doping testing kind of in everyday life in fitness centers?
So there are different ways to collect this DBS sample. You can either do a finger prick, that's the most traditional way of doing it, a more manual process, which requires some skills from the doping control officers and training by the doping control officers. But alternatively, you can use a more automated device, for example, a TASO device, which is manufactured in the US, which is easier for the DCOs to handle. So one of the things we actually wanted to examine before starting out was to receive feedback from both the athletes, but also on the DCO on which of these collection methods that they preferred. So was it the finger prick or collection with the TASO device from the arm? And it was quite clear from the study we did. We included 110 athletes that not only was it more convenient for the athletes to have the TASO device collection, but it was also affiliated with less pain compared to the finger prick. So that was actually the method that not only the athletes, but also the DCOs preferred. It was easier for them. And we also asked them whether they preferred over the urine or the conventional venous blood collection. And that was the case in 96% of the cases. The athletes preferred this new DBS method.
Oh, wow.
So that was quite convincing. So, of course, in order to implement the method, you need to write some guidance for the DCOs. We also made some videos for the athletes to get familiar with the method. And we also had to communicate it to the fitness centers and within the sporting environment. So they became aware that there was this new procedure coming forward. And as I mentioned before, we have to be clever when using a method, which for many of the substances that are detected in urine does have a shorter detection window in the DBS. So we really had to figure out how to take advantage of the method when testing in sports. So for example, we are able now to do, we call it mass screenings, meaning that we can come to an event, let's say a weightlifting competition, and with a certain amount of DCOs, they can maybe collect 30 samples from 30 different athletes instead of doing maybe 8 or 10 urine collection, which we normally do. So hopefully this kind of practices can also be a deterrent for the athletes so that many can be tested at once instead of not being tested.
Definitely. I think it's nice when all of the data and all of the practicalities sort of converge and point to the same solution. It sounds like DBS in a lot of ways is solving a lot of problems that come with this urine collection. Not only is it faster, so you're able to screen more, but it's also less expensive, I think, in a lot of ways than those urine tests as well.
The time of collection, we also examined that in our study, and whether it was the Tasso device or whether it's the finger prick, it took 2 minutes on average to collect the sample. And although it can take a bit longer, it's pretty sure that you get the sample within 5 minutes. And for the urine, if you come to a competition, the athlete could be dehydrated. It can take several hours. Of course, that's also a cost for us for paying our DCOs to wait for the urine to be provided by the athletes. So, of course, it gets a bit cheaper for us in terms of the salaries when we do the DBS collection. And there are other advantages also, such as shipping costs and other things to it. But we have to use it smart in our testing program, I think.
Absolutely. And I think very importantly, I'd like to highlight that Anti-Doping Denmark has had positive tests for samples collected via DBS. And I think this included even the world's first positive test with this method. So, Jakob, can you talk about the circumstances surrounding maybe these first initial positives and kind of what was going on and what you were thinking?
So I think we were one of the first organizations to actually implement it into our testing program. So we wanted to focus on, since we had this, we can call it the fitness DBS menu, where there's mainly anabolic steroids and other similar anabolic agents in our screening. We wanted to do it smart, so test in sport where you would benefit for taking anabolic. So it's mainly power sports such as weightlifting, powerlifting, but also some team sports. I just checked the numbers. So today we actually have 5 cases based on DBS. The first one was back in 2021, where the method was approved just before we got the first positive. It was in weightlifting, and it was a sample that was collected out of competition. So in a training environment where the athlete tested positive for 2 different anabolics, metanolone, stanozolol, and then an anti-estrogen called tamoxifen. So this was just a normal training control out of competition control in a weightlifting club. The second one we got, that was another weightlifter. He tested positive for a drug class called beta-2 agonist. The substance was terbutaline. This is actually something you take when you have asthma. But actually, there's been a different research project now coming out from Copenhagen University showing that it has anabolic properties also. And we talked to this guy, and it was not because he had asthma or anything. He actually got it from a friend because he recommended taking it for gaining muscle mass. So that was maybe a little bit surprising that it was actually also used for these purposes. The third one was at the World Championships in powerlifting, and it was actually a former world champion who tested positive for 2 steroids, metandinone, stanozolol, and also, again, the tamoxifen, the anti-estrogen. This was a competition, and we were kind of ordered from the International Federation to collect a certain number of urine samples mainly. But since it was on Danish soil, we also asked if we could be there and collect our own samples for our own program, so to speak. So we actually had DCOs sitting within the audience looking for suspicious athletes and then notifying them after the competition in their hotel rooms. And we were lucky there to get a third positive for these steroids. The fourth one was positive for trenbolone, which is also an anabolic steroid. It was a guy from the national team in American football. It was also out of competition control at a training venue. And then we have a 5th case just recently, which hasn't been made public yet. So I can't really go into the details on that. But that's quite exciting that we have quite a few cases now with the DBS method.
Absolutely. And I think that highlights, like you said, there is this deterrence effect, but you are actually detecting cases of positive tests where you might have missed them if you didn't have this DBS method that allowed you to sort of test more athletes and have this specific steroid profile that you're looking at. So then what is the process for Anti-Doping Denmark once you are notified that there has been a positive test, whether it's from dried blood spots or urine or other matrices?
It's kind of standard practice in every case, regardless of whether the substance is found in urine or blood or DBS. We always look up whether the athlete had one of these therapeutic use exemptions for taking the substance. So if the athlete has a health condition that requires taking a prohibited substance, we look that up in our system. And if that is not the case, and this happens, for example, for ADHD medication, asthma medication, stuff like that. But if this is not the case, we always call the athlete and we arrange an interview. And this is preferably a physical meeting, so the athlete can decide where he wants to meet us. And it's always myself and my colleague, who is an investigator, who meets with the athletes and prepared some different questions related to the substances and the circumstances and stuff like that. And sometimes we do some further investigations if needed, and then we hand the case over to our legal team. Then they recommend a sanction. And usually for these substances, anabolic agents, it's usually 4 years. And then it goes into a hearing process, which can either be written or oral, depending on what the athlete prefers. And if it is a oral hearing, we meet up in the sport court and defend our position. And then the case is decided within some weeks. And then, of course, the athlete has the right to appeal. And we can also appeal the decision if we're not in agreement with it. So that's a normal procedure.
Interesting. And is it the same for an elite athlete who's sort of a professional athlete, as well as someone who might have a positive case turn up when they're just working out at a gym recreationally?
The procedure just described is in sports. So it's everyone who's under the national anti-doping rules. So in fitness, it's a bit different. They receive a letter where we state that we have found the substance, and they have the possibility to reply whether they had the substance from the doctor as treatment, stuff like that. But there will not be a hearing as such. It's more decided in writing.
Well, that makes sense. And thinking about some of these positive cases, positive tests that have come out of the dried blood spot method, what has been the significance for you, Jakob, in being involved in this process and seeing those result in anti-doping rule violations?
So as I also mentioned, not only in sport, we have these 5 cases, but we also had a lot of cases in the fitness centers simultaneously. But of course, in sports, when we implement changes to our anti-doping testing program, the objective is, of course, to improve some part of it. We knew from our research that athletes liked the method. We had kind of examined that in our research project, and they preferred over normal conventional urine collections. And the same was the situation for our doping control officers. It has some advantages in terms of shipping, storage, compared to both urine and venous blood. But we also knew that maybe the detection time would be somewhat compromised compared to urine for some of these anabolic steroids. So, of course, it was really great to see that we actually got cases, because that was maybe our greatest concern, whether the detection would be too much compromised. So we didn't have any cases. So I think it's also a result of the close collaboration we have with the lab, who developed the testing menu, but also, of course, as I mentioned before, the sport that we're testing. And so with this menu, we are really focusing on powerlifting, weightlifting, and sport like that, where anabolics are more prevalent than in other sports.
Very interesting. And maybe taking a step back and thinking about the big picture, Jakob, how do you see DBS dried blood spot testing maybe changing the landscape for testing in elite sport going forward?
For us, it means that in certain cases, we can do more unpredictable mass screenings, for example. We have had some doping controls at certain competitions, weightlifting, powerlifting again, where previous years, we have done maybe 4 or 6 samples. Now we have gone in and almost tested all the athletes within the competition with the same manpower, with the same number of DCOs during the same time period. I hope it also becomes this deterrent for people to know that, okay, it is possible for everyone to get tested at an event. The requirements for shipping are not the same as for example, venous blood. So right now, water and others, they're also working on detection methods that are usually performed in venous blood. So if the research projects are successful, it might be in the future that DBS can substitute the venous blood collection when testing for certain substances. So that would be a really great benefit. And furthermore, I anticipate that the athlete will really, or we know that they really welcome this new procedure. So not only is it less intimidating and invasive, but it's also faster than the more conventional samples, urine and blood.
Certainly. And of course, beyond testing elite athletes with DBS, we mentioned that Anti-Doping Denmark has implemented this DBS testing in fitness centers and gyms. Can you talk a little bit more about the process that was involved in getting this all set up and maybe getting all of the gyms on board with this change? And how is that program going today?
As I mentioned before, it requires that we set up a specific menu in the lab. And then we had some contracts with all the gyms that had to be revised, because in those contracts, it was actually said that when you sign up for fitness centers, you can be subject to a doping control, which consists of a urine sample. So that kind of had to be changed. So it's now just a water accredited matrix that can be collected from the guys who goes or girls who goes into the gym.
Yeah, you future-proofed it.
Yeah, exactly that. So that was the first part. After validating the method in some way, checking the urine results with the DBS, and we actually could see that we were finding pretty much the same substances in the DBS after talking to the lab again. So we implemented it the 1st of January, where we moved to the DBS. And of course, there are so many practical advantages when you're out in the gym, also for our staff to go out there just to be able to do the DBS testing within 5-10 minutes, and then you are off again. So it has so many advantages, and that's where we are now. Now we, of course, also looking into the testing numbers we get and stuff like that. And at the end of the year, I think we have to see how the numbers look and whether we have to change strategy or do something different. So right now, I think we're doing quite well, and everyone are happy with the new procedures.
Certainly. Do you have any lessons learned at this point with just sort of the first 4 months under your belt of what went well and whether there are specific things that you'd want to improve?
Right now, we just had very good feedback for our staff members. They're really happy with the new method. So it goes really a lot smoother out in the fitness centers. We haven't really examined how the people in the gym think of it. We haven't really examined that. But so far, so good, I would say. So we haven't really have any problems now. We also changed our shipping method. So instead of using a courier, we can ship it through ordinary mail and stuff like that. So I think so far it's been really successful.
Oh, very cool. And have the gyms or fitness centers themselves provided any feedback?
No, not yet, but definitely we are in contact. It's a little bit outside my area, but I guess that we will receive feedback from them at some point, and it's important, of course.
Fantastic. Well, I think this has been a tremendous success so far. Like you said, it's still a process underway that you will be soliciting feedback and learning more as you go to maybe take a big look overall at how things went at the end of this year. And I know you're involved in other research projects as well there, Jakob, at Anti-Doping Denmark. So is there another project that you have in the works right now that you're very excited about and would like to tell us about?
Right now, we're testing out, not a DBS, but it's another similar collection device from Tasso where you can collect, instead of doing a venous blood draw, you can collect blood into a small tube from the upper arm. And we're looking into comparing, together with USADA, it's a project we're doing with USADA, comparing the blood markers that we measure for the blood passport in this matrix. So we're collecting from the athletes, it's again on a voluntary basis, and we send it to the lab and they do the more conventional analysis on the venous blood, because we also collect the venous blood sample, and then with this upper arm collection. So compare, and if that is as good as the venous blood collection in the future, maybe we can move to this kind of collection. And then again, as I said, we are also funding prevalence studies, which will be very exciting. We did that last year. We haven't received the result yet, but we are both giving money to a research group who will examine the prevalence in elite sport in Denmark, the prevalence of doping in elite sport, but also in the society as a whole. So we are right now in dialogue with these research group on how to design the studies. So this is, of course, very interesting also to have these measurements now and then to see also if we're improving our work. Is there more doping within the society in certain groups, or is it increasing, decreasing, and stuff like that? So that's what I'm really looking forward to, getting those results at some point.
Oh, wonderful. And we'll touch back on this first project that you mentioned, this looking at blood collection with the traditional full blood draw with the phlebotomist, getting that venous blood versus it sounds like collecting the capillary blood just from the surface, those small vessels at the surface, still using a wet blood sample, but with that kind of automatic collection device with a smaller tube. So do you have any thoughts on what you expect to see with that kind of comparison that you're looking at?
Yeah, hopefully it will. The results will be the same. I have to say and give credit to USADA and World Athletics because they already did a study at the Boston Marathon last year. So there's actually already a publication out there. But we had a discussion. I'm sitting on a water blood expert group, had a discussion, and we wanted to kind of validate the studies and having more labs involved, more anti-doping organizations. So right now we are getting the samples in, and hopefully we'll be as successful as what the first study showed.
Very interesting. And I think this prevalence study is critical as well. I think knowing the scope of the problem or just the scope of what people are using across the country, both in elite sport and in the general population, is really important to inform your anti-doping efforts. Do you have thoughts on what you might see in terms of prevalence based on any past data or just thinking about what you know anecdotally in your role?
Yeah, we actually funded a project after the Rio Olympics in 2016, where almost 4,000 elite athletes in Denmark were included. I think maybe 1,500 answered. It was kind of a special survey questionnaire technique, something called randomized response technique. And here, although the result is affiliated with some uncertainty, the best estimate was that 3% of the athletes in Denmark had been doping at some point. And since now we have funded this new study, we think it's really interesting to see whether that number has increased or decreased when using the same method. So 3% was the number before. For the society, we don't really know. We haven't really any previous good surveys on that. We will include or send out surveys to 150,000 probably Danish people. And we'll get a pretty good idea with a quite good power in those analysis to get an idea of how prevalent doping and specific doping substances are in the Danish population.
Oh, very cool. Well, listeners, we can look forward to these results coming out relatively soon. And I know in these scientific research projects, as well as in anti-doping in general, you have to work with a lot of different stakeholders and partners to be successful. So, Jakob, can you talk about how the scientific team there at Anti-Doping Denmark collaborates with different partners, including the Partnership for Clean Competition, academic institutions, WADA-accredited labs, as well as others in anti-doping?
PCC has been extremely important for funding some of the students here in Denmark for these fellowships and for providing funding for different research projects for the Danish academia. So that has been very, very important. We also have a strong collaboration within the Nordic countries. So we have now a Nordic science group. We have a Nordic testing group also that meets 3-4 times a year, discuss challenges, new projects, research projects. So we try to benefit from each other's knowledge also in that regard. We have a close collaboration with the different universities here in Denmark. We have some really strong research institutions on anti-doping research. And as already mentioned, the lab in Oslo is very important. It's been a great partner for several decades now. And they're always willing to participate and contribute to our research ideas and projects. So I think that's probably the most important collaborators for us.
That's wonderful. And I know you and the team there at Anti-Doping Denmark are working to solve some important problems on the scientific side of anti-doping, but oftentimes there are limitations, whether it's resources, funding, time, staff, just the feasibility of the kinds of projects that you would like to do. So if we took away everything that normally holds you back, what is the one problem, Jakob, in anti-doping science that you would most want it to solve?
Maybe it wouldn't have the greatest impact, but again, coming back from the background I have and the first studies I did both during my master's and my PhD on autologous blood transfusions. So getting a really good direct detection method for this kind of doping procedure, that would be fantastic. Right now, we have the biological passport, where you can look at these variations that happens when you do a blood transfusion, and you can use that as an indirect evidence. But if you really had a good direct method that could detect autologous blood transfusion for several weeks, a month, that would be fantastic.
Oh, I love it. Well, we'll think about this project and ways that we could potentially solve it. What do you see as the biggest barrier right now for getting that solution?
The problem is that it's your own blood that you're doping yourself with. So it looks like the one you already have in your body. When you take out the blood, something happens to the red blood cells. But the problem is that many of these cells that get bad when they're stored, they're pretty quickly removed from the circulation after reinfusion. So although we see some changes to the cell membrane and other things, those cells are gone within a few hours. So making the detection window very short. So I think that's the main problem. I don't know how to solve it, but hopefully there will be somebody out there to solve it. I mean, there's been research projects within this area for 50 years now or something like that, but we're still not there, unfortunately.
Still working on it. And I think a lot of these problems in anti-doping are really difficult to solve. And we appreciate all of the work that you are putting towards coming up with solutions for these real world problems. And if our listeners want to learn more about you and all the amazing work that you're doing with your team, what is the best place for them to go, or what should they do?
I think going to LinkedIn, I have a LinkedIn profile myself, and Anti-Doping Denmark has a LinkedIn profile. And then, of course, we have a website, Anti-Doping Denmark.dk, where you can look at our research project that we are funding, but also you can look into a lot of different other things, such as our doping cases and educational material and stuff like that. So that's the place to go.
Wonderful. Well, listeners, definitely get connected with Jakob. Check out that website to learn more about everything going on there at Anti-Doping Denmark. And Jakob, it's been such a pleasure to have you on the show today. Thank you so much for your time.
Thanks a lot for having me.
Well, it's been a pleasure to chat with you, and listeners, wonderful to have you here with us as well. If you haven't already, it would mean so much to all of us if you could take a moment to rate our podcast and leave a review on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.